right, so I'm going to try to do a few recordings of the PowerPoints from this chapter, all these chapters for this section, and I've kind of done some streamlined PowerPoints to try to hopefully go through it relatively quickly and talk about what's the most important thing in each case. So, chapter 10 is mainly to review the basic chemical nature of the gene or of DNA. We're not really putting in the context of the gene so much right now, but really the structure of DNA. And um, so first of all, we have to think about there's quite a bit of history in terms of how scientists figured out that DNA was the genetic material or is the genetic material. And the stuff that they had to think about when they did this was to think about, well, what if we how are we going to know the genetic material when we find it? What kinds of things must it be able to do? And so the genetic material must be complex because we know that the genetic material codes for, in some way, all the phenotypes of all the organisms on Earth. And so, in order to make complex brains and organ systems and to have all the information to tell an individual cell to develop into a complex organism, there has to be a lot of complex information that can be stored. Uh, we know that the genetic material is responsible for heredity. That's kind of the definition of genetic material. We know that from Mendelian genetics. The whole idea of Mendelian genetics was that genes are inherited in a predictable pattern, and that genes somehow code for phenotypes. But you need to be able to replicate that DNA. And we talked a little bit about how in the cell cycle you go from the G1 phase to the G2 phase, and in the process, G1, S, G2, we start with a chromosome with one chromatid, and then we end up with a chromosome with two chromatids. And so that's replication. And so whatever it is about DNA, we need to, and we also say, this sister chromatid is identical to that sister chromatid. So we have to some way be able to replicate the DNA faithfully. And then we need the DNA to code for, now we know it's DNA, and we need to get it to code for the phenotype. So there has to be some way to get this information out of the DNA and express it into uh, the phenotype. And then genetic material has to be variable. We now know that it's DNA, and DNA is subject to mutation. And if it wasn't subject to mutation, then it could never change, and we could never evolve. We would never have, So when we have chromosomes like this, where we have big A, and little a, the difference between those things is that there's been a mutation somewhere in the past, and you get a different version of the gene in this case. And you need to have mutations, and you need to have variation in order to have variations in phenotypes, to be able to adapt to different environments, and for evolution to occur. So, genetic material must be complex, must be able to replicate, must be able to must be able to explain how it gets expressed into phenotypes, and it has to be variable. It turns out the DNA answers all these things, but um, uh, we didn't really know that at the beginning, and it took Watson and Crick and Franklin. So this is Rosalind Franklin, and hopefully you know some of this story. It's a pretty long story, and it's a good story. Maybe I'll make you guys watch a movie um, on on this story because it's very interesting. So this is Watson. This is James Watson. This is Francis Crick, and they were really interested in trying to figure out what the genetic material was, and they had in their head, for various reasons, that it was probably DNA. Rosalind Franklin was working down the train route down in, I think she was in, they were in Cambridge. I think she was in London, and she actually did a lot of measurements on DNA, and came up with a very with some data that Watson and Crick were able to use to build this structure that's shown here. That's the actual structure of DNA that they built. There's a lot of controversy involved in that. People say that Watson and Crick uh, stole the information from Franklin, and it's not all that it's not that straightforward, but it's an interesting story. And if you ever get a chance, read more about it. But for right now, I want to mainly get to the point of getting you to just have a working knowledge of what the structure of DNA is. RNA can also be genetic material. So we know going in, certainly historically they didn't know all this stuff, but over it's probably 60 years of research. So DNA was figured out. So DNA structure was figured out in 1953, and now it's 2016. And so, however many years that is, it's 50 years, 63 years maybe, that since we've known the structure of DNA. And so, most organisms have DNA as genetic material. Some organisms, particularly viruses, you can have a virus that has an RNA genome. But so these are the guys that can hold genetic information. The genetic information gets expressed in the form of protein, but we don't think of protein as being the holder of or the storage molecule for that genetic information. So it's either DNA or RNA, and for now we're going to start thinking about DNA. All right, so we need to be able to think about the structure of DNA, and DNA consists of nucleotides linked together. Each nucleotide has three parts: a sugar, a phosphate, and a base. And you'll have to be able to draw out nucleotide structure in the online format. I'm not sure exactly how I'll make you draw that out, but in life in general, you should be able to draw out nucleotide structure. And so nucleotide structure looks like this. You have a sugar, you have a phosphate, and you have a base. And I'll show you the more detailed structure of this in a little bit. If it's DNA, if it's a DNA nucleotide, there's no oxygen there, so it's deoxy. And so the sugar equals deoxy ribose, which makes it deoxyribonucleic acid, or the individual parts are deoxyribonucleotides. And then the base can be A, G, C, or T. We will look a little bit at those structures. And then the phosphate, you can have, usually there's three phosphates on the structure. So that's that. I'm going to turn this up a little bit, just because I remember last time it was a little quiet. So hopefully that won't be too loud. All right, so this is what a nucleotide looks like: sugar, phosphate, and base. And we'll draw it out a little bit more in a minute. So here's what the sugars look like, and you should be able to recognize these different sugars. The idea is that there's 5 carbons in the sugar, so there's a carbon here, carbon here, carbon here, carbon here, and a carbon here. They're numbered 1 prime, 2 prime, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And this is ribose because it has an oxygen here. So if it has a 2 prime OH, then it's ribose. And ribose makes up RNA. If it doesn't have an oxygen here, it's deoxy because there's no oxygen there. And so it's 2 prime H, and it's deoxyribose. And deoxyribose is in DNA, so that's where the D comes from in DNA. And this is where the R comes from in RNA. The phosphate will get connected here or here, and the base will get connected here. and then you'll have a nucleotide. So these are just the sugars without all that stuff. It's just the sugars. So if we take all that stuff away, well, so here's just the sugar. It's a 5 carbon sugar. It's a pentose. It has 5 carbons. It kind of looks like it might have six because, but this one's an oxygen, so that doesn't count as a carbon because it's not a carbon. Here's the nucleotides, or not the nucleotides, but the bases that make up part of the nucleotide. And for whatever reason, I've never really thought it to be all that important to memorize the full structure of these things. Certainly, I don't know, at some point in biochemistry you might have to. But I think what I like to think of this when we're studying this stuff is to have a working knowledge of it. So, like, what do you really need to know when you're thinking about DNA structure and you're trying to answer questions about it? Memorizing the exact structure seems to be a little much unless you're thinking about biochemistry and how these things are made. Um, but what's important about these things is that these guys are purines and they have a double ring structure. So in general, they have two rings. These are pyrimidines and they have one ring. All right, and that'll be important in a minute when we start putting DNA molecules together. A and G are the purines and in DNA, and C and T are the purines in DNA, and C and T are the pyrimidines in DNA, and uracil is the pyrimidine in RNA. So you need to know that. You need to know that A and G are purines, and you need to know that they have a double ring structure. What we also know is that in DNA, we have base pairs. And A and T pair together, and G and C pair together. These dots are hydrogen bonds, and they hold the two strands of DNA together. And notice that this is a purine, and this is a purine, and this is a pyrimidine, and this is a pyrimidine. So a purine and a pyrimidine always go together. So what that means is if we have a purine pairing with a pyrimidine, you always have a big one with a small one. And then the overall diameter of the DNA is always the same, and that diameter happens to be 2 nanometers. But the key is if you have DNA molecule and you have A's paired with T's and G's paired with C's and C's paired with G's and T's paired with A's, because you always have a purine and a pyrimidine, you always have a big one and a small one together, and so the diameter of the DNA is constant, and that size happens to be 2 nanometers. So the fact that pyrimidines pair with purines is important for the overall structure of DNA. Well, there's a close-up view of a phosphate. So that's what a phosphate looks like. It's actually, it's always negatively charged. And so what we'll see when we start putting nucleotides together is they look like this in a big DNA molecule. And we'll see maybe there's an A sticking off of this sugar, maybe there's a T sticking off that sugar, and a G sticking off that sugar, and a C sticking off that sugar. We have a sugar phosphate backbone, they call this. And these phosphates are always negatively charged. So the reason it's called an acid, acids tend to be negatively charged when you put them in water. So an acid, so if this was phosphoric acid, it would have this structure. But in water, it gives up all its protons, gives up all its protons, and it becomes negatively charged. Now these two will be bound in the structure of the DNA, but you'll always have this one over here being negative. So that negative charge is always present from the phosphates. So putting nucleotides together, they kind of look like this, where each nucleotide has a sugar and a phosphate and a base. So here's our base. That's a purine. I know it's adenine because it has an A right here and it says adenosine. So adenosine, adenine, it kind of depends on exactly what state they're in for exactly what they're called. But we have a sugar, we have a phosphate, and we have a base. All right, so that's a single nucleotide. So these guys up here are purines. You can tell that because there's a double ring structure. These guys are pyrimidines. You can tell that because there's a single ring structure. And the difference really is it's just sort of a matter of whether or not you have an oxygen or a nitrogen group in various places. So again, I don't think it's overly important to memorize the differences exactly, but that's T, that's C, this is A, and this is G. Now they don't have them base paired at this point. They're just showing the separate nucleotides just by themselves. All right, so if you had to draw a nucleotide, what would you draw? Well, we know we need a sugar, and we know we need a phosphate, and we know we need a base. So the way I think about it is I start with a house. So you have the oxygen at the top, then you have a house, and then you have a little chimney. Then you label these starting on this side 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Then this has to have a phosphate on it. Now usually it has 3 phosphates on it when it's just kind of by itself. But we'll just draw with 1 for now. So that has 1 phosphate on it. We tend to start using shorthand and because it takes a while to draw out all those phosphates. So if we had 3 phosphates, we might draw it like this. So that carbon's still there. And we get 3 phosphates. And each of these phosphates really has an oxygen in the middle of it. But we draw it like that just to show that there are 3 phosphates on there. And so the P is actually representing the full phosphate, which is this. So that's a free phosphate. And just so you have to just kind of know that those oxygens are there. But usually that's acceptable. And then the base goes here. And let's say it's an A. So this would be an adenosine. And actually, well, I haven't shown you yet, but if it's deoxyadenosine, that means we have an H here and we have an OH here. So that's deoxy. If it was ribose, if it was an RNA nucleotide, we'd have an oxygen there. So let's get rid of that. So that's a nucleotide. Kind of an abstract version of it, or what I like to call a working model. So now a single-stranded dinucleotide, one thing that's important about nucleotides or DNA, so DNA, if we drew a big DNA molecule, we're going to be drawing a bunch of DNA nucleotides linked together, and then eventually they're going to be double-stranded. And so we need to be able to draw nucleotides linked together. So the nucleotide is a monomer, and that monomer makes up the polymer, and the polymer is the DNA. So we have to link monomers together to make a polymer. So how do we link them together? Well, we have one nucleotide, make it a deoxynucleotide. Doesn't matter what this is, we can make it an A. And then we have this phosphate here. And then we have another nucleotide here, and it has an OH and an H, and maybe that's a G, and it's got its own phosphate. And this one will probably have 3 phosphates. And this might have 3 phosphates too. Kind of depends on the exact situation, but if we're going to link these together, this is going to start with 3 phosphates. So we need to link those together. And when we link them together, what happens is we lose two of these phosphates. So two of these phosphates go away, and we call that, so two phosphates linked together go away, and they don't have any carbons in them anymore, so they're called inorganic phosphate. And sometimes they call this pyrophosphate. But we get rid of those two phosphates, and then the one that's remaining, I'll draw the whole thing out, is used to link the two nucleotides together. So then this is on that carbon, then you got this guy here. Yeah, that would be an A. So that's a single-stranded dinucleotide. This guy still has his phosphates here. And remember how these are numbered. 1 prime, 2 prime, 3 prime, 4 prime is that one, and then 5 prime is that one. So now we've drawn this molecule. We've got a phosphate on the 5 prime end, and we've got an OH on the 3 prime end. So if this nucleotide here, if this is the 5 prime carbon, then that's the 5 prime end of the molecule, and this is the 3 prime end. So these are just some names of the different nucleotides, and you name them slightly different things depending on what status, what state they're in. And so the the nucleoside is just the sugar and the base, and then when you add a phosphate, it becomes a nucleotide. And then there's like specific naming conventions for each of those things. We don't really worry about that. What we do, we will see them listed like this. So DATP, DGTP, DCTP, and DTTP. Those are nucleotides, all 4 nucleotides. And together, if we just want to lump these together, we'll call them DNTPs. And that'll be important when we talk about DNA replication and that sort of thing. And now on the previous page, I showed you nucleotides linked together, and we had a sugar and a phosphate and a sugar and a phosphate, and we had maybe a G and an A, might have been the other way around. But remember, this has a phosphate on it, this has a 5 prime end, and this has a 3 prime end. Sometimes we'll just draw that sugar phosphate backbone like that and draw a G and an A, but we'll still have a 5 prime end and a 3 prime end. So it's really, really important to understand the 3 prime and 5 prime ends of DNA molecules. That comes into play probably more than anything else. What's going on in 3 prime or 5 prime. Now I forgot to say here that these are linked together by that bond, which is a phosphodiester bond. So the nucleotide is the monomer, the polymer is DNA, and the bond that links them together is a phosphodiester bond. You have to keep that straight. We're going to talk about different kinds of molecules. We're going to talk about proteins. Proteins have different kinds of bonds. Amino acids are the monomer. Polypeptides are the polymer. Peptide bonds are the bonds. Here we've got monomers are nucleotides, polymers are DNA, and phosphodiester bond is the bond that links them together. Here, this is talking about monophosphates, and the way I drew these out is I put a TP in there because they're triphosphates. We almost always talk about them in terms of their triphosphate form. All right, so it's important to. So we've just looked at the DNA molecule, sort of just we've looked at a dinucleotide so far, but DNA is actually a double helix, and I'm sure you've all seen this kind of a molecule before. That's not a very good drawing of it. We can try again. Maybe if we make it blue, it'll look better. Something like that. So there's our double helix. The backbone, sugar phosphate backbone, is formed through phosphodiester bonds. The two strands are held together by um, hydrogen bonds between the base pairs. And one thing that I mentioned before, there's two base, so between A and T, there's two hydrogen bonds. So this dotted line means a hydrogen bond, which is not a covalent bond, and that's why it gets a dotted line. And it's important that it's a dotted line. It's important that it's a hydrogen bond because we need to be able to separate the strands of DNA molecules. So we need hydrogen bonds, and base pairing hold it together, and it holds these strands together. These complementary strands. So if you have one strand that has a particular sequence, and the other strand is going to have the complementary sequence. So T pairs with A, C pairs with G, G pairs with C, A pairs with T. So that word for that relationship is complementary. So they're complementary DNA strands. They're also anti-parallel. What anti-parallel means is that if one of the strands runs 5' to 3', the other one always runs 3' to 5'. So they run in the opposite direction of each other. And that's important. And when Watson and Crick and Franklin were trying to figure out the structure, for a while they tried to put the two 5' ends on the same end, and it didn't match up. And when they finally decided to flip them around, it made sense that they would fit together. And so here's kind of a view of the of DNA in its anti-parallel and hydrogen bonding nature. And so over here we can see, if you just look at the details of this, you can see that there's a deoxyribose sugar right here. So that's a sugar. There's a phosphate right here. There's another sugar, another phosphate, another sugar, another phosphate, another sugar. If this was the very end of it, then this would have an OH on it. So the 3, and that's the 3' end. So that's 3' and 5'. And then the other one is flipped around and going the other way. So this is now 5' and it's going 3'. And the 3' will always have an OH on the 3' end. This is RNA over here, just showing that it is single-stranded, and it also has, in this case, this is the 5' end. The 5' end always has a phosphate. This is the 3' end, but it's single-stranded, so there's no base pairing, no other strand. We've got U here instead of T. We got G, A, and C are the same. And here's a T over here. You can see that U's and T's are pretty similar. The main difference, well, there's a couple differences in there. But U is the pyrimidine that plays a role in RNA structure. You can see the two base pairs between A and T, or the two hydrogen bonds between A and T, the three hydrogen bonds between C and G. So if you recall what a hydrogen bond is, here we've got an NH. N is highly electronegative and pulls the electrons towards it. So this H is partially positive. This C double bonded O, the O has a high electronegativity, so it's slightly negative. And so this H will have a D plus, and this O will have a D minus. And if you think all the way back to, well, it's not D, it's really delta. But if you think back to chemistry and the chemistry of water, that's what a hydrogen bond is. So if you had water and it was forming hydrogen bonds with another molecule of water, it's because that's got a partial negative charge, that's got a partial positive charge, and then that's a hydrogen bond between them, which is a slight electrostatic attraction. So it's relatively weak, but when you have a whole bunch of them together, you get a strong bonding of the molecules together. So DNA has 5' end and a 3' end, and it's double-stranded, and it's anti-parallel. And the two strands are complementary, meaning they have complementary sequence, meaning that A pairs with T and G pairs with C. We can heat this up or unwind it in other ways, like an enzyme might unwind it. But the idea is that you can unwind this double helix by heat, and then you can cool it down and it'll go back together. When we heat it up, we call that melting. When we cool it down, we call it, or what we also call this, denaturing. And when we cool it down, we call that annealing. Sometimes you'll see that also called hybridizing. So hybridization is a term for the two DNA molecules sticking back together. So a lot of times we'll see in various technical applications, like if we're doing an experiment and we're trying to work with DNA, there's a lot of experiments that you've probably heard of different things, but you could take a little piece of DNA and it will stick to a target. So this could be a target, and we would call this a probe. And we can have one little piece of DNA molecule find another DNA molecule. I would like that to go away. Okay. And so here, this is called hybridization. So we'll see in some techniques that we talk about, it's very common to use one DNA molecule to stick to another DNA molecule, and we call that hybridization. All right, so what's wrong with this picture? You can't do that. That's got to be 3' and 5'. So everything's always got to stick in a complementary and anti-parallel nature. So it always has to be one going one way, one going the other. So 5', 3', and the arrow is always the 3' end, and this one's going 5', 3' that way. It's always like that, 100% of the time. Never anything different. All right, now we can heat this up, and depending on how much heat it takes to melt the strands, that's called the melting temperature. So we'll talk about that on the next little slide here. So melting temperature, as you can imagine, because GNC has 3 hydrogen bonds and A and T has 2 hydrogen bonds. If we have a molecule with a whole bunch of G's and C's, it's going to take more heat to separate. Because there's more hydrogen bonds. So we'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nucleotides. There'd be 30 H bonds holding that together. If it was A's and T's, if we had 10 AT base pairs, that's 20 hydrogen bonds. So it'll take less heat to separate those. So we can illustrate that idea with a simple graph where we're going to measure, on the x-axis, we're going to measure single-stranded DNA. And we can just measure, you can use UV absorbance using a spectrophotometer at a certain wavelength where single-stranded DNA will absorb that wavelength of light and double-stranded DNA won't. So if you start out with double-stranded DNA down here, and everything's double-stranded, and then we take the temperature and we increase temp, the DNA is going to start out double-stranded, and then it's going to start to become single-stranded, and eventually it'll all become single-stranded. So this is denatured now, and it's single-stranded DNA. You might have another molecule that starts out this way, and then it doesn't start becoming single-stranded DNA until later, until there's more heat added to the system. So of these two molecules here, we've got a lot of GC content or a lot of AT content. Which one is which? So this blue one, does that have a lot of A's and T's, or does that have a lot of G's and C's? Well, the way to think about it is it's actually starting to denature earlier, or it's denaturing at a lower temperature. So that means it's easier to separate it. So there's more ATs in that case, and there's more GCs in this case. So we can categorize a particular DNA molecule by what temperature does it take for about 1/2 of the DNA to be denatured. So that's this inflection point in the graph. That means 1/2 of the DNA has been denatured. So we call that the melting temperature. So Tm equals melting temperature. So the melting temperature is the point at which 1/2 of the DNA has been denatured. And then this would be the Tm of the other one. And so the Tm of the GC-rich DNA is higher than the Tm of the AT-rich DNA. And so that comes into play in lots of different applications when you're working with DNA. All right, so let's talk a little bit more about RNA. RNA is single-stranded. So the differences between DNA and RNA are DNA, RNA, double-stranded, single-stranded, has T, has U, has deoxyribose, has ribose. So those are the three main differences. But probably the most important difference is, well, this what's important here is that this has a 2' H. So if we look at the sugar, in DNA that's an H, in RNA it's an OH. That's the 2' position. This can be reactive. So we'll see when we look at RNA splicing and things like that, or the RNA can be more reactive than DNA. So DNA is always double-stranded, and RNA is single-stranded. DNA has T's and RNAs have U's. And one of the most important things is this difference here, that RNA has an OH and DNA has just an H. The other thing that is important with RNA, because it's single-stranded, it can fold up. You could have maybe some A's down here and some U's down here, and they can find each other and then just kind of loop out, and these A's and U's can pair up. And this is secondary structure. So RNAs, because they're single-stranded, can fold up into complex secondary structure. And we can write secondary structure as 2 with that little circle means secondary structure. And I'll show you a more complicated version, but just to kind of make some, so maybe you've seen tRNA before. So tRNA folds up into a more complex structure. And that's just because the base pairs can stick together. And there's lots of different RNAs, and they can all fold up into crazy things. But DNA always looks like this. No, well, not like that. Let's try to give it some kind of semblance of a helix. DNA is almost always double-stranded. Now, you can think of secondary structures of DNA, and there's different kinds of DNA. The bDNA was the original, sort of the classic structure, and that's the most biologically active structure, the one you find naturally most of the time. But there are a couple of other secondary structures. So there's bDNA, aDNA, zDNA, which have slightly different structures. Not too worried about that. Right now we're just going to worry about the main bDNA structure. And that's kind of what these are the dimensions. 2 nanometers wide. And the reason it's 2 nanometers wide is because we always have a purine and a pyrimidine that are sticking together. You have these different So we have 10 nucleotides for a full turn of the helix. So here's this helix. It's going around. It gets back to where it started. And then it goes around again. It gets back to where it started. Goes around. And the distance between, so that's called a full turn of the helix. There's 10 base pairs. In between each base pair here, there's getting all kinds of messages. Uh, there's 0.34 nanometers in between these bases, and so there's 3.4 nanometers between a full turn of the helix. But notice that we got a 5' end and a 3' end, and we got the other one's anti-parallel, and that's important. And also notice that there's always a 3' OH. That's important. The exact dimensions of the DNA, they don't come into play a whole lot when you're thinking about just kind of the basic function of DNA. When we start thinking about chromosome structure, that'll come back into play a little bit. But for the most part, we're mainly worried about. We usually think about DNA in simple terms like this. We always have a 3' OH on each end and a 5' phosphate. So that's what DNA looks like. These are the different forms of DNA. I don't know. They look a little bit different. There's not a whole lot of biological significance to these things. I suppose there is in certain, but it's kind of advanced. Really, we think more about the A form of DNA, but just know that it can form some other structures. And we can also see this base pairing in between strands, in between a single strand of nucleotides, and that's called a hairpin structure. It's much more common in RNA. So, but when that's what's called a hairpin, and that's where you've got maybe A's and U's, and we can throw a G in there for good measure, and then there might be C, A, U here. So what we have is intra, so it's within the same molecule, intramolecular hydrogen bonding, or base pairing. So base pairing means the different bases are hydrogen bonding together. Sometimes, so you can get just a hairpin like that. You can get what's called a stem loop. So that's a stem, that's a loop. And that means that there's base pairing here, but not in the loop. RNA molecules are much more complex. So we can see some of these hairpins and things in DNA, but it becomes more important when we think about RNA. So there's a stem loop in DNA. Now this can even happen when DNA is double-stranded. So RNA does it a lot more because it's single-stranded, but DNA can form loops like this. If there's some kind of a repeat structure in there where it can loop back on itself and stick together, it can form that kind of a loop. Sometimes you can have single-stranded DNA. Certainly when you're replicating DNA, there's going to be a point where it's single-stranded, and then one of the you can form those things more easily, and you can tell this is DNA because it's got T's in it. But RNA, well, that's just a hairpin. But RNA can do a lot more complex structures. So you can have, so you get your single-stranded RNA, and it can form. So like I drew a little bit before, it can form a bunch of stem loops. That should be looped over there a little bit more. So that's a tRNA, and there's a lot of stem loops. And we'll see like ribosomal RNA that we see in ribosomes gets even crazier. It's got all kinds of loops, maybe a little loop, then a hairpin, then another loop, and it gets really, really crazy. So you can get crazy secondary structures of RNA. Different kinds of RNA. So we've got messenger RNA, transfer RNA, ribosomal RNA, And as we get going, we're going to see more. There's snRNA, there's siRNA, there's miRNA. So there's lots of different RNAs in the cell, and this is kind of relatively new information over the last 10 years or 15 years. They've been finding lots of these things. So, but all of these things. So what's key about these things is that they're single-stranded, and they can form these crazy loops, these crazy structures. So there's an example of an RNA. Um, that's probably a ribosomal RNA. No, actually, this was like RNase P. So that reminds me, because RNAs can form these structures, they can do stuff. So this is an example of a ribozyme. So a ribozyme is an RNA enzyme. So RNA enzymes, well, enzymes are things that can catalyze chemical reactions. And if you think about a protein enzyme, whenever we think about protein structure, we think about this three-dimensional structure of a protein, and then the substrate can fit into a particular little space called the active site. So that's a protein enzyme that I'm sure you're familiar with. Um, Now, DNA can't really fold up and form an active site because it's always double stranded, but this RNA, maybe a substrate could fit right in there. So, because RNAs can form these complex structures, they can catalyze chemical reactions. And then another thing about that helps them catalyze chemical reactions is that they also have those available OHs that are sticking off. So, in RNA, it's single stranded, and you've got your bases sticking off there. But in these sugars, you still have your 2' OHs, and so those are available to do chemical reactions because that hydroxyl group is more reactive than a hydrogen that would be in DNA. So RNA is catalytically active, or it can be. And catalytically active just means it can do stuff. It can act as an enzyme, and that's because it's single-stranded. It folds into complex shapes, and it has a reactive 2' OH. So RNA is really complicated, and it actually it's more and more complicated the more we learn about it. So really, I mean that's like the most important thing about RNA is that it's expressed in a single-stranded form. It can fold up into three-dimensional structures, and those structures can then go and do things and act catalytically. Another thing while we're talking about DNA is that DNA can be modified, and this one of the most common modifications of DNA is DNA methylation, and this is related to gene expression. So if you think about a gene, I haven't thought about this for a while, but if you think back to earlier classes, you might have an enhancer, a promoter, and then some introns and some exons. We'll be getting into this a lot later, but this is our DNA, and this is actually double stranded. We don't usually draw it all out, but then it becomes RNA, so it becomes a single stranded RNA, and this process is transcription. And transcription is the first part of gene expression. So remember we said DNA has to be able to store information, be replicated, be able to mutate, but it also has to be able to be expressed. And then eventually this will become protein. And this is translation. But this whole process is gene expression. But methyl groups, oftentimes you'll find little methyl groups, and a methyl group is just CH3. So you can add a small methyl group to, and usually it's somewhere near this gene like this, and having those methyl groups there blocks transcription, so you can't express the genes. And so that's an important thing about DNA structure is that it can be modified. So you could have this same gene in one cell and it won't be methylated and it'll be turned on, have in another cell, it will be methylated and it'll be turned off. And this is the most common type of methylation, is 5-methylcytosine, and that's just, there's the fifth, I don't know where you start numbering these things, but apparently that's position 5, and that's different than 5 prime. That's not the same thing. It's just position 5 of this particular nucleotide. And so you have a C, and then you have methyl C. Well, I just kind of illustrated that, but we'll do it again. So we've got our gene promoter gets transcribed into RNA. So that's one. Let's say that's in a liver cell, and we need in this, and then this goes and becomes a protein, and we need to have that protein in the liver cell. Now maybe this is in the kidney cell. We don't need this gene. We need the gene to be turned off. So this, and we have a promoter. So the gene's still there. The gene's still the same, but now we've got these methyl groups. So a lot of times they draw them as these little lollipop structures, just to make it simpler. So we got all these methyl groups, and now, so whenever we transcribe, we need proteins to bind on there and transcribe the gene. Now the protein can't bind for some reason, so those methyl groups block it, and we no expression. So that's a really interesting aspect of gene expression, and we'll come back to that quite a bit later. But right now, I just wanted to introduce the idea of DNA methylation in the context of DNA structure, that you can actually have modifications of DNA structure in the DNA. All right, so I think that's all we need for DNA structure, and I will leave it at that.